Good afternoon. My name is Vandi Amasunabwangi, and I'm here with my classmate Joyce Peña, and we're from Willow International School of Mozambique, Maputo, Matola, and we're here to discuss a project that has a focus on the easiest way to monitor soil microorganisms. Soil microorganisms are indispensable decomposers on Earth and are also the basis to promote element cycling. In order to protect our environment and understand the soil we depend on, this study is about monitoring soil microorganisms, exploring the degree of similarity in two regions, and finding out the relationship between the number of microorganisms and external and environmental factors. By culturing microorganisms, we identified the dominant species of bacteria and fungi in that area. Sequencing dominant bacteria, we proved that they are both beneficial and harmful bacteria of the soil at the same time. Analyzing chart, we found that the two regions have the same dominant species. Drawing the line chart to compare the number and pH are similar in fluctuation curves. Continuing this experiment can help farmers in planting crops understand that the number of microorganisms change with season and the influence that these microorganisms have on plants. There are lots of microorganisms in the soil, including bacteria, fungi, virus, algae, and so on. According to our study, the soil microorganisms living in the area of 1 acre on the upper level of about 15 centimeters weigh between 0.5 to 4 tons. Structure and composition of soil. Soil structure is the arrangement of soil particles that become reunited certain shape and size clouds with the stability to overcome external damage. Soil structure can be used as a basis of identification of soil and reflects on the degree of maturation of soil fertility and local hydrology. The size and arrangement of solid particles of soil not only affects soil moisture, nutrient storage, and supply capacity, but also influences the gas exchange, heat balance, microbial activity, and root elongation. The soil is composed of solid, liquid, and gas. Solid includes organic matter, the minerals of soil, and the microorganisms. Liquid mainly means the soil moisture, and gas is basically the air present in the pores of the soil. These three form a unity, providing the necessary living conditions for the crop. That is the material basis of soil's fertility. The kinds of soil microorganisms. The soil microorganisms in nature mainly divide into microflora and microfauna. Microflora contains bacteria, actinocytes, and fungi. Microfauna contains protozoans such as amoeba, ciliates, rotifers, and nematodes. Based on the usage of carbon, it can also be divided into autotroph and heterotroph. Autotroph uses carbon dioxide, while heterotroph uses organic matter as a source of carbon. There are a variety of soil microorganisms, and only about less than 1% can be cultivated in the laboratory. According to the growth of crops and the production of human diseases, most of them are beneficial. The role soil microorganisms play in nature. Soil microorganisms play many different roles. Take bacteria and fungi as an example. Bacteria are non-nuclear prokaryotic microorganisms. Generally, the soil contains from 106 to 109 bacteria per gram. The features that bacteria play are that they can mineralize organic matter into a variety of elements. The nitrification, which is basically turning nitrate into nitrous oxide and nitrogen, increasing nitrogen source in soil, and they also contribute with the dissolution of phosphorus, iron, sulfate, manganese, and other components of soil. Among them, actinomycetes are also in the nitrification with non-ligand symbiotic, release plant hormone, and product antibiotics. The features soil fungi play are that they carry out the role of binding and mineralization of organic matter, release plant hormone, use mycelium soil binding particles to promote soil aggregate effect, produce antibiotics, and inhibit the growth of other microorganisms. The symbiosis of fungi and plant roots can promote plant absorption of nutrients and water. The changes of soil microorganisms. When external and environmental factors occur, they may cause a change in soil properties, which will have a direct impact in anaerobic, aerobic, and facultative microbial populations. Healthy soil has a high microbial diversity. However, when the soil environment changes, some microorganisms may stop growing, which will cause the plant to lack certain nutrients. In long term, crops may become the victim and the problem of global food storage may get even worse. Microbial diversity plays an important role in maintaining the high quality of soil, but now many studies on soil-related problems are still lack of understanding, such as the index of soil microorganisms and the soil microbial diversity and variety of microorganisms changes in the proportion of physical and chemical factors on the environment. In addition, that due to global warming and environmental pollution, the changes are likely to affect microflora. As we mentioned before, the research purpose of this experiment is to monitor soil microorganisms in campus and investigate the relationship between the number of microorganisms and external and environmental factors. The relation in the two regions in a relatively campus and to identify the dominant species of fungi and bacteria and the change of number through time. This experiment requires some equipment which are respectively autoclave, laminar flow, electronic scales, magnetic stirrer, petri dish, aluminum flask, thermometer, pH meter, glass probe. Shaking incubator, vortex tube, test tube rack, paraffin film, PCR machine, PCR cool box, and electrophoresis tank. It also requires some chemicals which are respectively nutrient broth, potato detrose broth, streptomycin sulfate, DEB, light matrix tube, vortex, chloroform, isoamyl acid, isopropanol, ammonium acetate, agarose, TE, TAE, and lithium bromide. Research method 1. Selection of sample area. We select two districts A and B, which are small trampled grass area under a coconut tree at a distance of 9 meters. Each of the sampling areas needs to have a size of 10 by 10 centimeters. 2. Collection of soil. We collect about 20 grams of soil for this experiment. 3. The pH measurement of soil. 4. Cereal dissolution. 5. Preparation of medium such as Na medium for cultivating bacteria and Pga medium for cultivating fungi. 6. DNA sequencing of strain. 7. Extraction of chromosome DNA. 8. Polymerase chain reaction or PCR. And 9. Gel electrophoresis. Result. We compare between A and B zone the temperature, pH, and rainfall. Based on our charts, the change of temperature and rainfall on PDA medium and NA medium have nothing in common, while the change in pH number on both medium has something in common. Results of DNA sequencing. We chose in total 9 different species for this experiment. We found that in terms of bacteria, the A zone contained B5, 6, and 9, while the B zone contained B1, 3, 4, and 8. And in terms of fungi, F1 was in B zone and F2 was in A zone. The relation between the change of dominant fungi, bacteria, and season. We denied dominant bacterium, the biological species which has a large coverage and frequently appears at the plate. According to the data, the dominant species of bacteria are respectively B1, which is Bacillus cereus, and B4, which is Bacillus micrans, and the dominant fungus is F1, Micropyrosis arundinalis. In conclusion, we found that the soil has a high microbial diversity and the two regions' dominant species have a lot in common. These microorganisms help allow with the cycling of materials in nature such as carbon, hydrogen, oxygen, sulfurs, phosphorus, nitrogen. And they also contribute with the biological decomposition of dead plants and animals. The creatures on the planet are influenced by the change of Earth's climate, and so do the numerous bacteria that play an indispensable role in the ecosystem. By monitoring dominant species in the soil, we found that microorganisms may be affected by environmental factors such as temperature, rainfall, and pH. Thank you.